300 million people, 300 million people worldwide are sufferers of type 1 diabetes. So far, there are no great treatments. There is no cure. But there is hope. This recent research, led by Doug Mountain, was able to create beta cells from embryonic stem cells that secrete insulin in response to glucose. This is a very important discovery or development. As we know, type 1 diabetes is caused by the autoimmune destruction of pancreatic islet cells. There's not much we can do about it. One treatment is to implant the cells of a cadaver, but unfortunately those are kind of hard to come by. And it's very expensive, and the treatment really lasts only 5 years. It's not available to everybody, but with this groundbreaking research, they have been able to make these cells in mass. And they paved the way towards a cure for this disease. My name is Liliana Rodriguez, and today I will be talking to you about exactly how they did this and how they were sure that they had found the answer. So, some previous research that was done, as you can see on this chart here, these are the different stages of cell differentiation in the experiment. This is a previous effort. But the problem is that these previous efforts were only able to get up to this point. They weren't able to get to the functional beta cell. And this is because the cells weren't able to secrete insulin. They didn't develop into adult cells. They didn't express the right markers for them to recognize that it was a beta cell, or they displayed other abnormalities. Now, what this group did was take these previous efforts and change them through a different set of differentiation methods, and to try to get to that beta cell that they were looking for. And the way that they did this was that they got tissue culture flasks with different groups of cells of the same kind, of course, embryonic stem cells, and used that previous method. But when they got to that point, they added in new things, and this was, of course, by guess. And once they got to something that resembled a beta cell, they then conducted ELISA tests with glucose to see the amount, if at all, of insulin that was being released by these newly developed cells. Alright, so in this next slide here, we see the results, or the findings. And this is of the in vitro. This is not on a live subject. Now, as you can see here, there's 2 control groups and the 1 test group, the newly developed beta cells. Now, the first control group is of the previous efforts, which we know don't work at all. The ones here are pancreatic cells from a working pancreas. So these are cells we know react to glucose and secrete insulin. And these here are the test beta cells, which they didn't know if they would work or not. And so what's being measured here is units of insulin being secreted, and on the bottom here is the amount of glucose that is being tested or given to these cells. Now, the reason that they did these different amounts of glucose in between 30-minute intervals is because they wanted to see whether the cells would just turn on when they sense glucose or whether they would just turn off. And this is because you don't want the cells to do that. You want them to be reactive to the glucose, and even if there were intervals. Because if they did it like that, they would not be safe to put into anything that was alive. And so, as you can see, the ones from the developed beta cells, the results, the amount of insulin being secreted, is very similar to that being secreted of the working pancreatic cells. Now, after they found that this was functional, they then went in vivo. So as we go back to the previous slide, they went and transplanted these functional cells into diabetic mice. Now, these are the findings from that experiment. So this is the same thing again, but we are now measuring insulin, human insulin that is being secreted versus mouse number. Now, as you can see again, there's the two control groups. The one with the working pancreatic cells, the one with the previous effort, the cells that are non-functional, we know those don't work, and then the test beta cells. Now, although the measurements are a little bit different, we can see that, again, the measurements are very similar to each other. And with this information, we can conclude that these cells that were developed in vitro are also effective in vivo. Now, what are the implications of this research? Well, a good one is that this research group, this group of people have paved the way towards an effective treatment and eventually a cure for this disease that is causing the suffering of over 300 million people worldwide. Another thing that is needed to talk about is the ethical question of this sort of research. People who don't know, for people who don't know, embryonic stem cells are harvested from human embryos, or babies that have not been born yet. This is an ethical question that everyone must decide for themselves. Do we use human embryo cells to try and conduct research like this to save 300 million people, or do we not do it? I hope that this has sparked some interest in stem cell research, if not awareness of stem cell research and the importance that it has to us as a society. Thank you very much for your time. Does anyone have any questions? I'm moving my fingers around too much. 8 minutes?